sekreterare för Nobelförsamlingen hälsar er välkomna till Karolinska institutet. As Secretary of the Nobel Assembly, I wish you most welcome to Karolinska institutet. Nobelförsamlingen vid Karolinska institutet har idag beslutat att Nobelpriset i fysiologi eller medicin år 2006 gemensamt tilldelas Andrew Fire och Mello för deras upptäckt av RNA-interferens, utsläckning av geners uttryck med dubbelsträngat RNA. The Nobel Assembly at Karolinska Institutet has today decided to award the Nobel Prize in Physiology or Medicine for 2006 jointly to Andrew Fire and Mello for their discovery of RNA interference, gene silencing by double-stranded RNA. Die Nobelversammlung am Karolinska Institutet hat heute beschlossen, den Nobelpreis des Jahres 2006 in Physiologie oder Medizin Andrew Fire und Mello gemeinsam für die entdeckte RNA-Interferenz, abschaltende Genexpression mittels des doppelsträngigen RNAs zu verleihen. L'Assemblée Nobel de l'Institut Karolinska a décidé ce jour d'attribuer le prix Nobel de physiologie ou de médecine 2006 conjointement à Andrew Fire et Craig Mello pour leur découverte de l'interférence ARN, inhibition de l'expression des gènes par ARN supplémentaire. Nobeliska Assemblea pri Karolinska institutet сегодня решили присудить Нобелевскую премию за 2006 год в области физиологии и медицины за место Andrew Fire и Craig Mello за их открытие РНК-интерференции, тушение приложения генов через двустороннюю РНК. We now distribute the press release. Please distribute that. And this year you get the press release in one piece. The first part is in Swedish, followed by a figure with Swedish legend. And then second part in that booklet is in English with the same figure with English legend. And those figures are the same as those figures which will now be used in presentation of the award. And the presentation is given by Professor Göran Hansson, Chairman of the Nobel Committee.
So what you just heard this year's Nobel Prize for Craig Mello for their discovery of RNA interference, gene silencing by double-stranded RNA. På svenska tilldelas alltså priset från de amerikanska forskarna Craig Mello för deras upptäckt av RNA-interferens, utsläckning av geners uttryck med dubbelsträngat RNA. I will begin the presentation in English. After that, we'll be happy to entertain questions in Swedish or English, or if we manage, also in other languages. No guarantees for any others. So what is RNA interference? It's a fundamental mechanism for controlling the flow of genetic information in living organisms. And let me introduce that concept. The genes are found in the nucleus of the cell. And here at high magnification you see the DNA molecules that contain the genes. The genetic code in the DNA is copied into a messenger RNA, or mRNA, which then is transported out into the cytoplasm, the blue part here, where it governs the synthesis of proteins. And these proteins are involved in all the life processes, as enzymes to get our food in the stomach, as receptors for signals in the brain, and so on. And you can imagine that it is critical to control this flow of information in such a way that the stomach gets its enzymes and the brain its receptors. 50 years ago, we thought that we knew all the important principles of gene regulation. And scientists were using genes to develop new medical therapies, improve plants and crops, etc. But unexpected results appeared in all kinds of organisms, from worms to plants to mammals, results that were difficult to explain with the knowledge at that time. It seemed that an important piece of the puzzle was missing. I'll give you an example from one of the many organisms where unexpected results occurred, and that's the case of the white petunias. Biologists wanted to make red petunia flowers even more beautiful. So the gene was introduced to make red color pigment. But this made the flowers lose color completely. And when they were analyzed biochemically, the messenger RNA for the, we call it color gene, had disappeared instead of, as was hoped, being increased. And here you see the sad example. Here's a normal petunia that you can all recognize. And here is one where they tried to enhance the red color and instead got a white color. And here is kind of mixed result. So, what happened to the mRNA in the greenhouse and in many other laboratories where similar problems occurred? The explanation came from Andrew Pyer and Craig Mello and Semorhabditis elegans, this little worm that had turned out to be so useful in biomedical research and was also involved in the Nobel Prize 4 years ago. This is Pyer and Mello's key experiment. They were studying a muscle protein that helps the worms move smoothly. And they made a synthetic type of messenger RNA, here called sense RNA, which was injected into the worm. Nothing happened. Then they made a mirror image of the sense RNA, an antisense RNA, and injected it. Again, no effect. But in a third variant, they mixed sense and antisense RNA, which then bound to each other in the test tube. And now they injected this double-stranded RNA. And the double-stranded RNA made the worms twitch, lose their smooth movement and start to twitch in a similar way as worms that had a defective gene or muscle protein. So Pyer and Mello concluded the double-stranded RNA can silence genes. And it silences the gene whose code matches that of the RNA. So it's a highly specific protein. And they could also deduce that gene silencing involves enzymes in the cell. They published their findings about genetic interference by double-stranded RNA in the journal Nature in February of 1998, so a little bit more than 8 years ago. And that started an intense research for the components of the RNA interference machinery. And now we understand the basic principles and components. Double-stranded RNA, when it gets into the cytoplasm of the cell, is bound to a protein called dicer, which cleaves it into a smaller piece. The two strands separate, and one strand of RNA binds to another protein, an enzyme called RISC. And this RNA piece helps RISC to bind to a messenger RNA that has the same sequence as this one. And that RNA is attacked by RISC, and the messenger RNA is destroyed. So if we go back to the flow of information in the cell, it's interrupted.